Hello, and welcome to our webinar, an improved IDT solution for generating large CRISPR HDR knock-ins, introducing Alt-R HDR Donor Blocks and Alt-R HDR Enhancer V2. My name is Molly Schubert, and I'm a staff scientist in the Molecular Genetics Research Group at IDT. I will be serving as the moderator for today's presentation. Our speaker today is Jessica Woodley. She is a research scientist in the Molecular Genetics Research Group at IDT. Jessica earned her master's degree in microbiology from the University of Iowa, where her research focused on Staphylococcus aureus biofilm formation. Since joining IDT in 2016, her work has focused on the development of RHPCR technologies and improved methods for efficient homology-directed repair in CRISPR applications. During and following the webinar, please submit your questions using the question box. Any questions that are not answered during the Q&A session will be followed up with afterward. All right, Jessica, I'll pass it off to you now.
All right, thank you for that introduction, Molly. Now today, I'll be walking you through some work we've been doing for the last few years to improve HDR rates and streamline the HDR design process. Now, as Molly mentioned, this webinar will highlight two of our newer CRISPR products, including the Alt-R HDR Donor Blocks and the Alt-R HDR Enhancer V2. I also want to let you know that we'll have a discount offer available to you at the end of the webinar for the new Alt-R HDR Donor Blocks, so stay tuned for that. So to start off with, I want to do a quick review of the DNA repair pathways. So after you've used your CRISPR enzyme of choice to generate a double-stranded break, the predominant repair pathway is going to be non-homologous end joining, or NHEJ. And this will result in non-specific insertions and deletions, or indels. And this is generally useful for generating knockouts or conducting genomic screens, and is generally high efficiency. Now, if you include a repair template or donor DNA with your CRISPR reagents, you can then get repair through homology-directed repair, or HDR, and this will result in the generation of specific mutations or insertions. And so this is very useful if you want to generate a specific allele through SNP changes to mimic disease, or if you want to generate a gene fusion such as a GFP tag. Now, the main point of our talk is considerations for improving the HDR efficiency in research. And so when you consider the two repair pathways, NHEJ is very predominant. And so one of the biggest challenges for HDR is just improving that overall HDR efficiency. And so the first portion of the talk will focus on how using improved HDR reagents can enhance your HDR rates. And so we'll focus on a couple different ways you can use modifications of the repair templates to generate those higher HDR rates, and also how you can use small molecules to modulate repair pathways. The second portion of the talk will focus on using improved designs, including just general CRISPR design elements, such as your guides, as well as focusing in on the donor itself. So here's a look at IDT's full workflow for CRISPR research, going all the way from experimental design through analysis. So I'm just going to focus in here right now on the HDR reagents. And so for small edits, we do have our Alt-R HDR donor oligos available. So these are single-stranded DNAs that are up to 200 bases, and they are modified to improve HDR. We have previously also had our megamer long single-stranded DNA available for larger knock-ins, and these go up to 2 kb and are sequence verified via NGS. Now, our two new products, which are highlighted here, include the donor oligos. And so these are linear double-stranded DNA that are modified to improve performance. They go up to 3 kb, and again, like the megamer, are sequence verified via NGS. And then finally, we have that small molecule enhancer, our V2, which has been really game-changing for improving our HDR rates. So for several years now, we have recommended the use of long single-stranded DNA, our megamer, for large knock-ins for HDR. So I'll get into the reasoning behind that in just a moment, but there are several key improvements offered by the donor blocks that I'd like to highlight. And so here, I just have a table comparing our plasmid offerings to our unmodified linear double-stranded DNA offering. focused on our high-fi G block. Again, our long single-stranded DNA or megamer, and then our modified linear double-stranded DNA, which is the donor block. So one key thing that comes up right away is the overall length of the donor blocks. The megamer only goes up to 2,000 bases, whereas the donor blocks will go up to 3 kb. In addition to that longer length, it also allows for more flexibility for sequence complexities such as GC content or repeats. So if you've run into that in the past with megamers, hopefully you'll have an easier time with the donor blocks. Um, in addition, we do have a slightly faster turnaround time with the donor blocks relative to the megamers. So that's starting at 17 to 21 business days. Some of these will be faster, but generally the time to required to manufacture these is dependent on many factors, including complexity and sequence length. So that's why we have a little bit of an asterisk there. Um, these donor blocks are also a more cost-effective solution. So you can see we are going up to a 10 microgram scale. So 3 micrograms is great for microinjection, but is a little limiting for cell culture work. And so we are going up to a higher yield there. And you can see here that the prices are also cheaper for both yield options relative to the megamer. And lastly, the modifications added to the HDR donor block um, offer improvements in HDR efficiency and particularly in accuracy over the unmodified double-stranded DNA. So taking a step back now, we'll discuss why we focused on a modified double-stranded DNA template. So when we consider potential repair templates for large knock-ins, one of the easiest to produce is plasmid. And so that's one of the great things about it. However, overall efficiency is pretty low with plasmids in our experience and what we've heard from the field as well. And that's generally tied to the toxicity to cells. So because cell viability is so low, you're really limited on the amount of plasmid that you can deliver. Um, uh, in general, if you then consider linear single-stranded DNA, such as our megamer, it really works well for HDR, and as long as you control the dose, uh, it has pretty low toxicity to cells. Um, However, the main drawback for long single-stranded DNA is the cost and time for manufacturing these. Because of the more complicated process to produce these, um, it's really limiting on the yields that we can deliver. And so the 3 microgram yield that we offer is great for microinjection work, but it's not a very cost-effective solution for cell culture work. And so we began looking into alternatives and turned to linear double-stranded DNA. And so we've generally found that linear double-stranded DNA works well for HDR, um, and it's easier to produce quickly and at large scale. However, its main drawback is the risk for off-target integration. And I'll get into that in just a moment. But based on reports in the literature, we then asked if modifications could be used to reduce this risk of blunt integration with double-stranded DNA so that we could offer a more cost-effective solution for large knock-ins. So here is our model of repair when using a double-stranded DNA template. So again, after you've used your CRISPR enzyme to generate a double-stranded DNA break and introduce your linear double-stranded DNA donor, ideally you'll get a nice clean repair through HDR. However, because these blunt ends can also be recognized by the NHEJ repair machinery, this linear double-stranded DNA can actually get blunt ligated into the double-stranded DNA break. And so when this happens at the on-target site for Cas9, it results in the duplication of one or both of the homology arms at the on-target site. And we're referring to this as homology-independent integration or blunt integration. Um, and generally at the on-target site for large templates, this event is fairly rare, and you can also screen for it to make sure that you're getting the correct repair that you want. However, the real risk comes from the fact that this can also occur at Cas9 off-target sites or at endogenous double-stranded breaks. And so unless you're looking for that, you could miss it, and then it could have unwanted effects downstream. And so our solution to this has been by adding modifications to the ends of the donor or within the homology arms, we can prevent this homology-independent integration. And so we've done this by incorporating modifications into universal terminal sequences on the end of each donor. And so that's roughly 30 base pairs on each side. This modification pattern helps increase the HDR rates and then also mitigates the risk for these unwanted blunt integration events, both at the on-target site and at potential off-target sites. In addition, these universal sequences are not incorporated into the HDR edits and do allow for more consistent synthesis with that faster turnaround time. All right, so here is a sample from our screening. So we sent out to screen a wide variety of modifications for this ability, and this is just a sample from those results. But our general approach to this was to introduce modifications into a short double-stranded DNA donor that contained a 42 base pair insert with an EcoRI restriction site. We would then deliver this donor with Cas9 complex to either the on-target guide RNA or a guide RNA targeting an alternative locus, thus allowing us to create a mock off-target double-stranded break. So after editing, we then amplified the target sites and assessed insertion using EcoRI digestion. And then based on the sizes of the digestion products, we were then able to quantify the HDR rates and the blunt insertion rates. So um, in general, in this experiment, we were looking for modifications such as G. So you can see in green here, we get an increase in the HDR rates relative to the unmodified donor. You can see we get a nice decrease in blue in the blunt insertion rates as well at both the on-target and off-target sites. And we get a nice boost to the ratio of HDR to blunt rates as a result. So once we had identified our top modifications in our screen, we then set out to further confirm them with additional cell lines and at additional sites. And here's a summary of our results shown here. When we were testing these shorter sequences, we found that our top modification pattern here, labeled as Alt-R mod, improved the ratio of HDR to blunt insertions at the on-target site up to 4-fold. All right, so before moving back into larger knock-ins, I do want to take a second to pause and introduce our second new CRISPR HDR product, so the Alt-R HDR Enhancer V2. And so, like I said, this V2 enhancer has been a really game-changing improvement for us. So after we had launched our original HDR enhancer, which is shown here relative to other small molecules in light green, you can see that it gives a decent boost relative to DMSO or untreated control cells. Um, and it works well in many of the other published compounds that we looked at. However, after we had launched this product, we did continue evaluating new compounds, and we have found several new solutions that improved HDR above the original HDR enhancer. And so our top small molecule solution is presented here as the HDR Enhancer V2. In addition to offering the best improvement to HDR out of everything we have tested, V2 can also be used at a lower concentration. So it allows for better cell viability due to less DMSO being present. All right, so here I want to highlight, once we had identified our best modifications for the Alt-R HDR donor blocks, we then went into looking at how it behaved with large knock-ins relative to other alternative templates. And so here we're comparing long single-stranded DNA, shown in gray and light blue, versus an unmodified double-stranded DNA product, and in green, and our Alt-R HDR donor block. And so we're delivering these donors into K562 or hex cells at equal molar doses. Here I'm showing cells that have not received any additional treatment. And so you can see for generating these GFP insertions that the long single-stranded DNA gives roughly 5% HDR as a baseline. If we then switch to the unmodified double-stranded DNA in this case, we're getting up to about 9% HDR, and use of our modified donor blocks boosts that up to about 15% HDR. And if what's interesting is if we use long read sequencing to dig further into these editing events, what we found is that uh, the long single-stranded DNAs did result in these partial integration events where one junction is not fully integrated. So you can see here with the non-targeting strand, the right junction is not incorporated all the time. And with the targeting strand, the left junction is not being incorporated all the time. In contrast with the double-stranded DNA donors, we did not observe these partial integrations. What was really nice to see is that the treatment with the V2 enhancer was very beneficial for the long double-stranded DNA templates. So now if we add this V2 treatment, um, you can see that the single-stranded DNA HDR rates were brought up to about 10 to 20%. However, with the HDR donor blocks, you can see that went up to 50 to 70% HDR. So the use of the Alt-R donor blocks and the enhancer V2 really offers an improved solution for large HDR insertion events. Okay, so the final bit of data I'm going to share in this portion of the talk is focused on assessing how the Alt-R modification helps reduce the risk of homology-independent or blunt integration into the genome. And so here we're focusing more in on the insertion into a non-targeted double-stranded break. And so in this experiment, we're using those same GFP donors and assessing them at a potential off-target that we've generated by combining the Cas9 with an alternative guide RNA. So there's no homology in this case, and this actually represents the worst case scenario where the off-target double-stranded break is generated at near 100% efficiency. So if we compare the blunt insertion rates of an unmodified donor versus our donor blocks, you can see that the unmodified double-stranded DNA for these 4 different loci is actually generating about a 25% blunt insertion. By adding our modifications, you can see that this unwanted integration is brought down to about 8%. And what's really nice to see is if you use the V2 enhancer, which should be inhibiting the NHEJ pathway, does bring this further down to basically background detection levels. All right, to recap this section, the use of our Alt-R modified HDR donor blocks does allow for improved HDR rates and does inhibit this unwanted non-homologous integration of a double-stranded DNA donor template. And then we can further improve this by adding our small molecule Alt-R HDR Enhancer V2, and that will further push things towards HDR and inhibit these unwanted integration events. All right, so moving into the second portion of this webinar, we'll be discussing how improved HDR designs can increase HDR. And so we'll be talking first on the selection of high activity guides and how that's really important, and how the placement of the HDR edit relative to the cut site plays into effect. Focusing now in on the design of the donor, we'll also discuss the use of blocking mutations to prevent recutting after HDR, and a little bit on optimal homology arm lengths. And so all of the data in this portion of the talk is going to focus on Cas9. However, we have done extensive testing with Cas12a as well. And so there are resources for that on our website and in a recent publication in Scientific Reports. All right, so in general, I do want to say these experiments are first going to focus in on the use of single-stranded DNA templates, our short donor oligos, and then expand out into the donor blocks. But in our first step in designing HDR experiments is always going to be guide selection. And there are 3 major factors to keep in mind here. First is the position of the cleavage site. Generally, you want to be as close to the HDR insertion as possible. Second is guide efficiency. You want to have a high efficiency guide to mediate high efficiency HDR. And you also want to consider the risk for off-target effects. So choosing a guide with a good specificity score will help reduce the potential for off-target editing. So in general, you can use in silico tools such as our Cas9 design tool for addressing some of these concerns. But first, we'll be walking through a case study highlighting some of these considerations, such as the positioning and guide efficiency. And so in this case, we are inserting a 6-base EcoR1 restriction site right before the stop codon of the GAPDH gene, shown here. And I've highlighted several PAMs that are within the region. So you can see we have some guides that edit very close to our target HDR edit, and then others that will generate a double-stranded DNA break within 15 bases. All right, so as we look at the HDR rates here, it turns out that position really isn't everything. So the guide that cuts 6 bases downstream from the insert is actually the guide that gives us the highest HDR rates. So you can see if we look at this by EcoR1 cleavage, we're getting over 50% HDR. And in contrast, you can see that the guides that cut only 2 or 5 bases away from our desired edit are actually really poor efficiency guides. And as a result, it really does limit the HDR rates. And in fact, the guide that cuts 14 bases away is actually giving us the second highest HDR rate because it is also a high efficiency guide. And so this is actually an extreme case where the editing rates are lower, but it does highlight the importance of confirming your guide activity prior to HDR experiments. All right, so here we're just highlighting the effect of insert location relative to cleavage site. So we are looking at the serpency1 locus in this case, and again, inserting an EcoR1 site right at the cut site or up to 27 bases away from the cut site. And you can see, as long as you're close to the double-stranded break with your HDR insert, rates are pretty high. So as long as you're within 10 to 15 base pairs, you should be able to get decent HDR. But it really does drop off once you get further away from the cut site. And unfortunately, you may run into a situation where guide availability is limited in your sequence of interest. And so what we wanted to know is if you're outside of this optimal window where you can get good HDR, how can you improve your HDR rates? And so one of the most important factors we found with these edits at larger distances is strand selection when you're using a single-stranded oligo as your repair template. And so generally what we found is that for insertions that are near or at the cut site, the preferred strand does vary. So you'll have to empirically determine which strand gives you the better HDR rates. However, if you are outside of that recommended 10 to 15 base pair window, it really matters which side of the cut site you're on relative to the PAM. So in these experiments where we were inserting, again, an EcoR1 site, 25 base pairs upstream or downstream from the cut site, you can see in the case of the PAM distal orientation, so on the opposite side of the cut site as the PAM, that the non-targeting strand is actually the preferred strand pretty dramatically. In contrast, if you insert on the PAM proximal side, the targeting strand is generally strongly preferred, which you can see here. I will note that these donors did have silent mutations incorporated to prevent recutting, which I'll get into on the next slide. All right, so we then further investigated whether there were any preferences in the placement of the silent mutations for these types of HDR edits. So in this experiment, again, we're inserting a sequence 25 base pairs upstream or downstream, and we decided to compare donors that had no mutations, that had a PAM mutation, or that had repair track mutations that were placed on the same side of the double-stranded DNA break as our HDR edit. And what we found is that you really get the best HDR improvement when those mutations are incorporated on the same side of the double-stranded DNA break. So in the case of the PAM distal edits, these repair track mutations gave the best benefit to your HDR rates. In contrast with the PAM proximal PAM proximal designs, a PAM mutation alone was really sufficient to get that good boost to HDR. In this case, we tested against 3 genomic loci with a small insert placement. And as I'll show in a moment, we have confirmed this trend with larger inserts as well. So in this experiment, we are inserting a 300 base pair sequence, so pushing the limits a bit more here, 20 base pairs upstream or downstream from the cut site. And again, we've compared donors that had no mutations to donors that contain PAM mutations or that contained the repair track mutations. And so first, if we look at that 300 base pair insert placed directly at the cut site, you can see when we treat our cells with the V2 enhancer, we're getting up over 50% HDR. If we then switch to either the PAM distal or PAM proximal insertions, you can see without any mutations that those HDR rates are dropping dramatically since we're outside of that 10 to 15 base pair window. And again, what we see is we get the best HDR rates when our silent mutations are placed on the same side of the double-stranded DNA break as our HDR edit. So the track mutations were most beneficial for the PAM distal designs, and the PAM mutation alone was sufficient for the PAM proximal mutations. All right, so the last bit of data in this section is going to be looking at optimal homology arm lengths. As with everything, I'll be showing you some data from our single-stranded donors. So if we look at 3 different insert sizes here, you can see HDR is plateauing pretty dramatically with 40 base pair homology arms. Anything longer isn't really benefiting your HDR rates, and that's because those modifications are really providing some protection there. Now, if we switch gears, we did want to assess this with larger inserts as well. So this is some data with our HDR donor blocks. And so here we're testing a 120 base pair insert, a 500 base pair insert, a 700 base pair insert, or a 2 kb insert at a variety of genomic loci. And these donors were delivered as either equal molar doses or equal mass doses. And so we decided to test this because in our experiences, we generally find that cell lines will be limited on the total mass of donor that you can deliver before you're impacting your cell viability through the innate sensing of double-stranded DNA. And so the exact amount will vary by cell lines, but we typically try to deliver as much material as possible for higher HDR. And so the doses that we used are indicated here, generally around 1 microgram being the equal microgram dose, and the equal molar doses varying from 25 up to 100 nanomoles, depending on size. And so if we compare homology arm lengths of 40 up to 500 bases, you could see with our short inserts, so the 120 and 500, that the HDR rates are really kind of plateauing, particularly for the equal mass dose, around 100 to 200 bases. In contrast, if we look at our larger inserts, the HDR rates really aren't plateauing until you hit 200 to 300 bases. So overall, we generally recommend starting with around a 200 base pair homology arm length, and that is the default in our design tool, but this may vary by cell type or system. All right, so in conclusion, as you're focusing on your designs, we do recommend selecting a guide RNA with a high editing efficiency that is placed as close as possible to your desired HDR mutation. And it's a good practice to check your guide RNA editing potency prior to HDR experiments for the highest likelihood of success. In cases where your guide availability is limited and you can't get an edit that's close to your double-stranded DNA break. Um, Generally, you should be careful when you're using your single-stranded DNA templates and select the appropriate strand uh, according to whether you have a PAM distal or PAM proximal insertion. So with PAM distal, you want to use a non-targeting strand for your single-stranded DNA. And with PAM proximal insertions, you want to use a targeting strand. And in general, you want to use silent mutations that are placed on the same side of the double-stranded DNA break as your HDR edit. For homology arm lengths, we recommend starting with 40 bases for short insertions and 200 to 300 for your long insertions. And in general, whenever your HDR mutation is not sufficient to prevent recleavage, you should include silent blocking mutations to improve your HDR rates, whether you're using an HDR donor oligo or an HDR donor block. And if you're looking for a streamlined way to design your donor DNA, we have developed an Alt-R HDR Design Tool based on experimental data that will help predict the best guide and donor template combinations for your research. All right, so with the last bit of time here, I do want to highlight our HDR Design Tool. So it uses a friendly UI and is based on empirically defined design rules. And is fully integrated with our Cas9 guide RNA design tool as well. So as I said, our design tool was based on empirically defined rule sets. I just want to highlight a little bit of that here. So in this particular experiment, we generated a SNP just upstream of the PAM. And so we looked at the impact of mutating every other base within the PAM and seed region to every other possible base in several cell lines and at several sites. And we use this data to inform the selection of blocking mutations for our Alt-R HDR design tool. And generally, you can see within each position, there's not a huge preference for the particular base, but you can see that generally mutating the GG of the PAM or mutating right around the cut site offers the best improvement to the HDR rates in these cases. All right, so I do want to give a little bit of a look at our HDR design tool. And so the first step in using the design tool is picking the targets. And so the design tool does allow a very flexible input. So you can use a gene or genomic coordinates to select in human, mouse, rat, C. elegans, or zebrafish. And if you do so, it does allow you to select an NCBI transcript that will automatically upload any coding information for incorporating silent mutations. Um, if you're working in another species or would like to do batch designs, you can also use FASTA inputs. From there, you can then specify your designs with these easy to use sliders. And so you can specify the region that you would like to either insert a sequence or introduce SNPs. So here in this example, we are generating a CAT mutation to an ACA. And you can see down here in the menus, you can either use our default homology arm length settings, which are either 40 or 200 bases, depending on your length. And you have the option of including silent mutations. And you can see there is also a preview of the coding region here, so you can verify that your mutations are doing what you would like. And then in terms of designs, it will generate a list of donor and guide combinations. So you can see these are visualized here. In our example, we're including a design that does not have any silent mutations added versus one that does have silent mutations added. You can see it's very easy to read with our guide and cut site indicated, as well as all of the mutations that are introduced. And here in the case study for these two particular designs, you can see that we do get a nice boost to the HDR rates with those silent mutations. And this is all easily integrated with our website ordering, so it allows for a one-click reagent design to order. All right. Um, so, in conclusion, uh, if you are trying to improve your HDR rates, um, we do offer several different products where we have introduced modifications into our HDR donors to improve HDR rates. So we've got our HDR donor oligos that are available, as well as our HDR donor blocks. We also have our HDR Enhancer V2, which is an exciting new small molecule solution for boosting your HDR rates. Um, and finally, you can really do a lot to improve your HDR rates through improved design, whether it's the selection of the right guide RNA and CRISPR enzyme, as well as HDR designs. So using your silent blocking mutations to prevent recutting and improving your insertion frequency. And so again, our Alt-R HDR Design Tool really helps streamline that process to give you the best potential outcomes. All right, so I do want to highlight we have further resources on this. If you would like the links to any of these products, they're listed here. For design support, again, here is the link to our design tool. We also have a publication focused on short HDRs with Cas9 and Cas12a in scientific reports. And for the large HDR section, we also have an application note on design optimization. So a lot of the data that was presented here will be present in addition to a couple other things. In terms of delivery, we do have protocols available both for our donor oligos and for our donor blocks. There is a link here. And this will give you some nice starting points for recommended doses of the donor blocks to use for your best outcomes. In addition, you can reach out to our scientific support group if you have additional questions or need any additional support. And with that, as I mentioned at the beginning of my presentation, we do have a special offer for webinar attendees. So please visit the link here to save 30% on Alt-R HDR Donor Blocks. This is a one-time single purchase promotion of up to 5 Alt-R HDR Donor Blocks. So we think this is a great product and we'd love for you to try them out. With that, I'd like to say thank you for listening, and we'll move on to the Q&A now. So I'll pass it off to Molly.
Thanks, Jessica, for your very informative presentation. All right, so it looks like we've received several questions from our audience. Okay, so the first question here, have the HDR donor blocks been tested in mouse embryo injection? And I guess we have some other questions too about what other systems that they've been used in.
All right, that's a great question. So through alpha and beta testing, our HDR blocks have successfully been used for HDR in mouse embryos, zebrafish, C. elegans, a variety of immortalized cell lines, iPSCs, and primary Ts. We're also frequently asked about the V2 enhancer, so I'll address that here as well. The V2 enhancer works in multiple cell types, including primary cells and iPSCs. It's not typically used in embryo injections, though, due to the concerns over the DMSO content.
Sure. It looks like we have a question here more about the design of the donor template. So there's a question about is there any benefit to using asymmetrical homology arms with the HDR donor blocks?
That is something that we've looked into a little bit. We haven't really seen any benefits to asymmetric designs ourselves, so we typically stick to that 200 to 300 base pair length as I presented. However, if you do run into complexity issues with your genomic sequence, asymmetric designs could be a good way to get around that. As long as on the shorter end, you have at least 100 base pairs of homology or so.
Sure. Here's a good question. So someone here is asking if we have any delivery methods for liposuction.
That's another good question. We do have that formal protocol available for Lonza electroporation. That'll give you recommendations for volumes to use for your cells, for your donor RNP, as well as guidance on amounts to use for those reagents. in terms of micrograms to avoid any viability concerns. If you reach out to our scientific support group here, we've got that email address listed here. We do have additional support for alternative delivery methods like the neon electroporator and also liposuction. I will say that the HDR rates for large knock-ins with liposuction were a little bit limited, but we'll start you off with a good starting point, at least based on our internal testing.
Great. All right, here's another question. So someone is asking here, does IDT offer larger sizes of the HDR donor blocks?
That's a good question. And the 3 microgram and 10 microgram yields are our standard offering. We've frequently gotten asked about larger yields as well. And for that, you can contact us directly, emailing our genes at idtdna.com group, and then they can discuss with you other options.
Great. Well, unfortunately, it looks like we've run out of time to answer additional questions. If you do have additional questions, please enter them into the Q&A box and we will contact you directly if they weren't answered here today. Again, I would like to thank Jessica for her time here with us today. And lastly, thank you again for attending, and we wish you the best of success in your research.